From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on October 11th, 2023. Hello, Micronauts, and welcome to our Quality Quorum. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 11th episode of Matters Microbial. Thanks as always for listening and or watching. First, a denizen of my Wunderkammer. Now, I have to tell you I adore fossils, and trilobites are among the very, very best of them. And I've been collecting them for some years, and the idea of these arthropods swarming around in ancient seas 500 million years ago has always fascinated me. I had never collected a trilobite of my own, however. I simply bought prepared specimens. So through social media, I got to know a very serious fossil hunter named Marcus Martin. That's quite a name, isn't it? Who owns Beecher's Quarry in upstate New York. There, special conditions during the fossilization process create amazingly detailed trilobite specimens, including the soft spots and soft parts that are not seen in most fossils. The pyrite formed during fossilization creates what Marcus calls gold bugs. My wife and I were fortunately able to travel to this famous quarry a while back, and as you can see, I was very excited to be there. I was even able to split out this fossil as is. And then Marcus was able to prepare some of my finds professionally. And here is a tiny gold bug that he prepared seen at a distance. And here's an amazing close-up. You can really imagine these tiny arthropods swarming about in those ancient seas. And I have to tell you, I was beyond happy. I will put a link in the show notes to Marcus's work, which is worth your time. Today's inspirational lapel pins. Growing up, I was very influenced by George Orwell, and Animal Farm in particular. The lessons it taught me are always near and sometimes far too topical. I was also fascinated by Robert Louis Stevenson's The Strange Case of Dr. Jekyll and Mr. Hyde. And the quote from Dr. Jekyll, "You must suffer me to go my own dark way," still gives me chills. But then it's October and getting closer to Halloween. 7th week of my microbiology class continues unabated. My micronauts have followed up on their marine microbiological safari. Observations by micronauts remain key. The best equipment in the lab is this one. And so the students were able to find yet another wildly fluorescent marine bacterium seen here, what very well may be a red fluorescent marine microbe here, and this absolutely amazing bicycle wheel colony. The structure of bacterial colonies continues to fascinate me. My micronauts also learned a bit about antibiosis, as you can see here. The red bacteria appear to inhibit the growth of the surrounding bacteria. This is the kind of thing that we hope to find among microbes associated with lichens. Finally, many strains of the bacterium Serratia make a bright red pigment called prodigiosin, but they only do so at room temperature, as you can see here. When the bacterium is grown at 37 degrees C, the red pigment is not made, leading to white colonies. So I had my micronauts take those plates with the white colonies and incubate them for a week at room temperature. And lo and behold, you can see prodigiosin being made at the perimeter of each colony. This was such a great opportunity for discussion with my micronauts about that and what it might mean, especially to regards what takes place within a colony. Many years ago, through comparisons of DNA sequences, the late great Carl Woese discovered what some people thought were bacteria, but turned out to be a third domain of life, the Archaea. Now, I find it's a challenge to talk to Archaea or talk about Archaea to students because they're so used to thinking about eukaryotes like themselves or bacteria. But fortunately, the Giant Microbes company actually does make a tiny plush archaeal keychain, so there's hope. And who better to chat with us about his adventures with extremophilic Archaea and their equally extreme viruses than Dr. Ken Stedman of Portland State University in Portland, Oregon. Welcome to our Quality Quorum, Ken.
Hi, Mark. Glad to be here.
Delighted to see you. So would you mind if I ask a kind of a question first? And I guess the one thing I wanted to tell everyone, and I'll put this in the show notes, is the name of your lab is?
We are the Extreme Virus Lab. And if you're looking for us online, extremeviruses.org.
It's absolutely wonderful. I love the pictures. I love the information there. And I thought it would be a wonderful place for people to start. And I'll put a link in the show notes. But I think the first question, just in case we have listeners or viewers who aren't familiar with the Archaea, how would you define them?
So the Archaea are really fascinating microbial organisms. And one of the things that I think people shouldn't forget, they're also microbial eukaryotes. So one of the things to think about, we've got bacteria, we've got Archaea, and we've got eukarya. They're the big eukarya, like you and me, but there are also lots of very small eukarya. And these are incredibly understudied, but way more diverse at a molecular metabolic level, et cetera, than any of these, as some of my colleagues like to say, charismatic megafauna. So the Archaea themselves, if you just look at them, and looking at them even with a very high-powered microscope, they end up looking a lot like bacteria. They've got rods, they've got cocci, et cetera. But as soon as you get to a molecular level and start to look at how the actual biochemical and molecular biology mechanisms work on the inside of these cells, they're actually much more like you and me than they are like other bacteria that they look like. And so that really fundamentally differentiates Archaea from bacteria. And one really fascinating thing that we found in the last few years really is that we probably have common ancestors with the Archaea more recently than the Archaea have common ancestors with bacteria. And so if anything, the Archaea are our cousins much more so than they are to bacteria, which was a huge discovery and partly why Carl Woese is very well known. And I got involved working on this because while I was taking not a microbiology course as a student, but I was a teaching assistant for a microbiology course that was taught by my PhD advisor, the amazing Sydney Kustu. She introduced Archaea, and I thought to myself, wow, these are really cool, or really hot, as we'll get into a little bit later. And that was what convinced me that I really wanted to work on these organisms. During my PhD, I'd worked on bacteria, Salmonella typhimurium in particular. But then for my postdoc, it's, let's go and look at these Archaea.
Oh, they're awesome. And I'm going to say something, and I know you'll probably disagree with me. I think, and maybe this is something that you've actually done a little bit of, and it needs to be showcased. One of the reasons we know as much about bacteria as we do is because of E. coli, which everyone is familiar with. It was easy to grow, easy to work with. And it would be nice if we had the equivalent among the Archaea. And actually, the one that you work on is probably pretty close to it, do you think?
So yeah, that's a great point. What is the E. coli of Archaea? And people will definitely argue about this. I have my own opinion. Other people have their opinions as well. In fact, Giant Microbes has an opinion. They think that it's Pyrococcus furiosus, or the flaming fireball, which is exactly what you have there. My favorite organism, as far as the E. coli of Archaea is concerned, is an organism called Saccharolobus sulfatericus. And it's very confusing because it used to be called Sulfolobus solfataricus. And so most people talk about Sulfolobus. I still have trouble doing that. And microbial taxonomy is something that I'm sure we could spend lots and lots of podcasts talking about, but we won't, not for today. Basically, the reason that I really like Saccharolobus is it's a hyperthermophile, and actually not really hyperthermophilic, depending on your definition. It grows optimally at 80 degrees Celsius, but it's also an acidophile. It grows optimally at pH 3. So 80 degrees Celsius and pH 3. That to me, at least coming from my background, which actually is not really in biology. I was a chemical engineer as an undergraduate. But everything I'd learned about biology says, 80 degrees Celsius, pH 3, your DNA falls apart, your proteins fall apart. This shouldn't be something which harbors life, or let alone something that actually particularly prefers to be under those kinds of conditions. Now, despite the fact that Saccharolobus likes to grow at 80 degrees Celsius and pH 3, it's actually relatively easy to work with in a lab. It's not an anaerobe, so you can actually grow it under atmospheric oxygen conditions. It's our heterotroph. You can feed it media a lot like the media you feed E. coli. We used to joke when I was in my postdoctoral lab with Wolfram Zillig, is basically all you need to do is take LB, lysogeny broth, not to be mixed up by anything else. It's lysogeny broth. And I heard this from Giuseppe Bertani. Joe Bertani himself said it's lysogeny broth. You take lysogeny broth, you pH it to 3 with sulfuric acid, and you heat it up to 80 degrees, and the Saccharolobus just love it. That's not exactly right. They need a few other things. But basically, that's the case. And so we can grow it very easily in liquid media. We can also grow it on plates. Now, plates have a bit of a problem, however, at 80 degrees Celsius. And I'm sure all of your micronauts knows, like, wait a minute, agar doesn't do very well at 80 degrees Celsius. In fact, it's kind of liquid at 80 degrees Celsius. So we use something called Gelrite. It's also been used quite a lot by plant biologists, also known as phytogel, which sets up really nicely, and you can actually grow these plates up to about 90 degrees Celsius. They stay relatively stable, unless your pH is a little bit too low, and then you hold up the plates to look at them, and then you have this hot liquid which slides onto your hand, which is a little less attractive. But generally, we can work with Saccharolobus a lot like you would work with E. coli in liquid media. You can work with them on plates. Again, you can grow them aerobically. They have one really big advantage, however, relative to E. coli, is all you have to do is put them on the lab bench, and they stop growing, because you're already at 55 or maybe even more than that degrees Celsius below the temperature they like to live with. So you don't necessarily need a fridge or a freezer. Just, literally pop them on the lab bench.
This is really a lot of fun, because a million years ago, I worked for a company in San Diego, and one of the products we made was from what we then called Pseudomonas elodea. And the exopolymer it makes is, in fact, gel-an gum. And that's what Gelrite is a derivative of it. So yeah, I used to make the stuff.
Yeah,
so
very cool.
It's really cool stuff.
Yeah, it's great. It really makes our life a lot easier, because being able to blow things on plates allows us really to do a lot of this kind of standard work with the viruses, which I'm sure we're going to get into in just a second here, which are then things that we grow on plates just like you would use with bacterial viruses, in terms of seeing plaques, in terms of being able to isolate them, look at their phenotypes on these plates. So having that Gelrite is really critical.
Before we move on to viruses, which is, I know, the focus, I just wanted to tell you that I wish there was a collection of exercises with Archaea. And I'm sure you've thought about that kind of thing. And I think it would really help. I mean, I hate to say it needs more microbial PR, but they really do. Because the number of students who talk to me about them, and they have like rote things that they say because that's what they were taught. So the idea of bringing it in early would be so important. And especially because, non-pathogenic, a lot of cool things you could do with it. Yeah. So I hope you and your folks are working on that.
We are definitely working on that. Now, one of the things I mentioned, Saccharolobus, the only sort of downside as far as working with these in the lab is, 80 degrees is actually pretty toasty. And having incubators that work at those kinds of temperatures and can hold those temperatures, unfortunately, tend to be a little on the expensive side. Yeah. And so another nice organism to use, which I didn't mention, are some of the what are called extreme halophiles. And so these are things that grow at very high salt concentrations. These actually grow at basically the same temperature, many of them, as E. coli does. And so some of the halobacterium, some of the haloferax organisms, and in fact, some of the genetics, particularly of some of the haloferax organisms, are much better developed than the genetic systems are in Saccharolobus. So if I were going to be setting up a course to work with some of these extreme halophiles or any Archaea at all, I would actually start with these extreme halophiles. The other thing is they have really pretty colors when you grow them on plates because they have kinds of pigments, some of which are really important for them being able to allow them to grow. And in fact, if you ever, I'm pretty sure you have, flown into the San Francisco Bay Area, what you do is you fly over these amazing salt pans, which are all kinds of different colors, and many of the colors there are due to some of the halophilic archaea, which are living in these salt pans, and some of the only things we can actually survive at those very, very high salt concentrations. And one fun aside here, when I was a teaching assistant for the microbiology course that was taught by Sidney Kustu, we actually had a field trip where one of the students' friends had a small plane, and we got to fly around the South Bay and call this a microbiology field trip, which it absolutely was, because we were going out and looking at the microbes. So this is a way that, yes, they may be very, very small themselves, but their effects can be huge.
Exactly so. Well, I got you going not about viruses, and maybe we should turn to that.
Well, let's do that because, of course, that's what we work on. And as we saw here, we're talking about viruses from hell. Well, what do we mean by viruses from hell? And to be perfectly honest, that is not my name, and it's also not a name that Mark Martin came up with either. This is actually, the viruses from hell is the title of an article that was written by a science journalist, Charlie Petit, in U.S. News and World Report, when he came with us on a field trip and was writing an article about the viruses that we were working on. And so why viruses from hell? Yes, okay, they're high temperature, low pH, but the particular place that we were looking for these viruses in, and there's a picture here of that particular site, is called Bumpus Hell. Now, why is it called Bumpus Hell? This is because an explorer in the 1800s in the Lassen Volcanic region, this is before Lassen Volcanic National Park became a national park, he was leading tourists to this thermal area in the winter, and unfortunately broke through the snow that was on top of some of these hot springs, and then into one of the hot springs himself, ended up surviving, but lost both of his legs. So this region now is called Bumpus Hell because of that. So viruses from hell, well, in this case, they're viruses from Bumpus Hell. And it was hellish for Mr. Bumpus, but actually it's a great place for these viruses to be. And so I'm sure you're just about to ask me, Ken, why do you study these viruses? Since Saccharolobus by itself is a really fascinating organism. Well, the reason we study the viruses, multiple different reasons. Viruses are amazing, and I guess we can kind of call them microbial a little bit, at least the viruses that are infecting microbes, and even the viruses that are infecting eukaryotes as well, they're also microbial. So the reason that I got really interested in studying the viruses that are infecting particularly Saccharolobus is because viruses have had probably billions of years to evolve ways to take advantage of their host cells. So if you want to learn something about a host, and this is actually true for lots of just general molecular biology, you study a simple system which you have a better chance of being able to understand. So I got interested in the viruses of Saccharolobus because I was interested in understanding the Saccharolobus and how on earth they could survive at these very high temperatures and low pHs. So to do this, you look at the viruses, because this is how we learned a lot about molecular biology, how we learned a lot about E. coli, how we learned a lot about us, is in the studying the viruses. So to learn about Saccharolobus, you study the viruses that are infecting Saccharolobus. And one of the first big surprises that we had, and in fact, this was even before I started working on them, this was the work of Wolfram Zillig, my postdoctoral advisor, who was one of, as they like to call it, Woese's army, for Carl Woese, supporting the fact that Archaea really were very different from bacteria. He started working on these viruses. In fact, some of the very first viruses and some of the very first biochemistry and molecular biology was on studying those halophilic archaea I was telling you about from the South Bay of San Francisco. Wolfram Zillig showed that the RNA polymerase, which is the molecular machinery that takes DNA and makes an RNA copy from it, is much more similar in archaea to us than it is to any of the molecular machinery in bacteria. So again, this is at the very molecular level, but what about the viruses? And some of you may have seen this funny little thing over my shoulder here. That's actually an image of our favorite virus. So this
is Stanley, right?
This is Stanley, exactly. Stanley the SSV, Stanley version 1.0, actually, because we now have a better structure of Stanley. But this is a 3D printed version of the structure of our favorite virus. And most of you notice, yes, okay, very strange looking virus. It doesn't look like that kind of virus up there, just over my finger, your classic bacterial phage. It also doesn't look like all the scary pictures you've seen of SARS-CoV-2. This is very different. And it's very different in terms of its shape. So we're very interested in how it makes this shape. So a lot of the people in our group are looking at the shape of this virus and trying to figure out how different pieces fit together. We've also done a very genetic approach to try and understand how each of the individual pieces then in the genome of the virus fit together in order to make this particular virus and allow it to infect the Saccharolobus. And one of the things that I also like to say, if Saccharolobus is the E. coli of Archaea, I like to think that SSV is the lambda of archaeoviruses. And it turns out it does a lot of things a lot like lambda. It integrates its genome into the host genome. The virus production is induced by UV irradiation. So lots of things are very similar relative to lambda, but at the same time, none of the genes look like lambda at all. So many things about this virus are really fascinating, and we really don't understand a lot of them. And so this particular model is a 3D printed version of our SSV, the Saccharolobus spindle-shaped virus. So this particular one is the first one to be found, originally from Beppu, Japan. This particular model is about 7 million times life size. So behind me on the shelf, which you may be able to see, there are a couple of viruses printed out there. I printed all of these virions at 1 millimeter to 1 nanometer. So they're literally 1 million times life size. And so if this were 1 million times life size, it would be about this kind of it's about 90 nanometers in length, which would be 90 millimeters in terms of 1 million times life size. So that's the structure here. Now you'll notice there's this wonderful tail at one end. So this particular tail, we think, but we don't know, is what binds to the Saccharolobus host cell. So this would be analogous to the spike protein that all of us have gotten, hopefully all of us have gotten injections in terms of the vaccine that then will help stop SARS-CoV-2 from binding to our cells. And so here it looks as if this tail structure is involved in binding to the host cell. Unfortunately, or fortunately, depends on how you look at it, we don't know exactly whether that's the case. And so we're trying to figure out exactly how these are interacting with each other. We think it's something special on the outside of the cell called the surface layer or S layer, but we still need to do some more work to really nail down whether these are actually specifically interacting with each other. So if you think about, the virus versus the host, you'll see my hand here is sort of representing the host. So the virus itself gets about 90 nanometers in length. The host Saccharolobus is about a micron in size, so 1,000 nanometers. So it would be about 10 times as big as this one particular virus particle. So actually, these virus particles are actually really pretty big, relatively speaking. And it turns out that the genome on the inside, that double-stranded DNA, is only about 15,000 base pairs. So it's relatively small. So what is it about this really big virus particle and the small DNA? Is that something about high temperature, low pH survival or something else? We don't know. So we have more to do. Back to the lab. So we're very interested in how all of these things fit together. And one of the things that we've been able to do at Portland State University, which I'm incredibly proud of, is to now take understanding this virus genome and put it into an undergraduate course. And so our undergraduate course here, it's called officially Advanced Molecular and Cell Biology Research Lab, but we just call it Mutant Viruses from Hell. Because what we do is we make mutant viruses in the mutant viral genome and then test it through the rest of the term. And so the students start out with mutants. We have no idea what those mutants are. And through the term, they then figure out where the mutation is, make some predictions about what that mutation could do, and then test to see what's actually going on. And so that's been incredibly successful. We've had hundreds of mutants which have been characterized, many of which have been published. And so I'm very, very proud of that particular process. And there's one other thing that I did want to mention actually as far as this process is concerned, because we have lots of pictures here, including on my website. You may wonder why are all these pictures of these really amazing hot springs on the website? Well, it turns out that there's actually a method to our madness. And that is, we're doing all of this mutagenesis to try and figure out how the virus works. But it turns out that nature has done this for us. So if you go to places where you have 80 degrees Celsius and pH 3, basically volcanic hot spring environments, you will find not only Saccharolobus, but you'll also find viruses like SSV. And the advantage that you have if you find the viruses like SSV in various different hot spring environments is that you can now compare all of these viruses to each other. And the ones that you find, these have been selected for by the magic of Darwinian evolution to actually be something which is successful. So the reason that we go to a lot of these places and collect samples, and you can see in these images us collecting samples in Lassen Volcanic National Park, collect these samples, bring them back to the lab, and then try and find viruses that look like this particular one and compare them to each other. And that gives us really profound insights into how these viruses are functioning, how they actually can survive at these very high temperatures and very low pH environments.
That's quite a story. And I especially like that undergraduates are involved in it. I mean, a lot of times undergraduates think that they can't get involved in things like that, and you're proof that that's not true.
Yes, and in fact, if you look at this picture now, it's a collection of the Extreme Virus Lab. And if you look at this picture, half of the people in that image are undergraduate students. We also have a couple of high school students who are involved in the process as well. So we're really very open to undergraduate researchers, getting them experience doing research, preferably also getting them out into the field. And we have this big advantage that where we go is incredibly beautiful and allows us then to have not only a great scientific experience, but also a great social experience in the same process.
Oh, I think it sounds lovely. I didn't have opportunities like that, so I think applause is merited for you and your students.
Thank you.
I think you have another type of virus that is especially interesting to people if they've ever heard about the term the RNA world. Would you care to talk a little bit about that?
So I'd be more than happy to. And actually, this is a nice connection between what we're finding in Lassen Volcanic National Park, high temperature, low pH environments, and things that we really absolutely did not expect. So one of the things that we've done, and lots of pictures on our website, and in fact, this picture right now here is a place called Boiling Springs Lake. Boiling Springs Lake is probably my favorite hot spring, at least the one that I've gotten to so far in the world. It's a really pretty amazing location because it's a lake, which is probably about 100 meters by 200 meters. The low temperature in the lake is 50 degrees Celsius. The high temperature is 94, 95 degrees Celsius, which is boiling at the approximately 2,000 meter altitude that it's located at. And the pH is about 2.5. So just crazy, crazy environment. This Boiling Springs Lake would be perfect conditions for Saccharolobus and for these viruses that you would find in that environment. Turns out we're completely unsuccessful in finding viruses there. Probably because there's no Saccharolobus that actually happens to grow in that lake. There are other organisms that they have to be able to infect. But we were very fortunate to have gotten some funding to do what's called a metagenome. Now, everybody knows about genomes, sequence the genome of this virus, sequence our genome, etc. Metagenomes are sequencing an environment. So we did a sequence of the virus-sized particles. So we filtered out all the archaea, filtered out all the bacteria, filtered out all the microbial eukaryotes, and just got the virus-sized particles from this high-temperature, low-pH ecosystem. And then we did sequencing on them. And at that time, I won't mention how long ago it was, we got only 400,000 sequences. Now, these days, 400 million sequences is what you'd expect. But in 400,000 sequences, what we found was something very, very strange. First, actually not strange at all, we found sequences that look like our favorite virus. So, great. So they're there, we just haven't found them yet. The other thing that we found, which was very bizarre, is that even though we had sequenced DNA, we're finding all these sequences that looked like RNA viruses. And even more bizarre than just RNA viruses, these look like RNA viruses that were infecting plants. Now, plants can't survive very well at 50 degrees Celsius. So what's a sequence that looks like an RNA virus that infects plants doing in a lake like this? So to make a long story short, what we found out was it was actually part of a DNA virus, now a very small DNA virus, that replicates through what's called a rolling circle system or a single-stranded DNA virus. Most of our DNA viruses that we think about now these days, Lambda, Mpox, are double-stranded DNA, like our genome. And actually, SSV is also a double-stranded DNA virus. But here you also have single-stranded DNA viruses, and there are a number of viruses, particularly those that infect bacteria, that are known to have single-stranded DNA that uses for their genome, and also a number of things. Some of you may know about the parvoviruses, which will infect dogs in particular. Puppy parvovirus, really nasty stuff. Vaccinate your dogs. But this single-stranded DNA virus, somehow, the one that we found in the lake, seemed to have picked up a gene that was otherwise similar to RNA viruses. So somehow these things are recombining with each other, and we call these the cruciviruses because there's basically been a crossing over between DNA and RNA. Now, we never heard of this, and in fact, we tried to publish this, and it got rejected from a couple of journals, which we will not name the names of those journals. But then when we finally did publish this, what we found was I kept getting emails from number of people have been doing metagenomics and saying, oh, thank goodness you figured out what was going on. We'd found these sequences and just completely ignored them because we didn't know what to do with them. And so we didn't need any more projects in the lab. This was still plenty to do with this particular virus. But this was a project, DNA and RNA viruses recombining with each other. Whoa, okay. So we need to figure something out more about it. So about half of the Extreme Virus Lab now is actually working on these kinds of viruses, the Cruy viruses, and then the other half is working on these SSV viruses. And one of the problems we have, unfortunately, you notice I've got a nice 3D model of this one. We'd love to have a 3D model of these Cruy viruses, but we don't have the virus. So far, we just have the genome, and not just a genome from Boiling Springs Lake. We now have hundreds of genomes of these viruses that we found all over the place, including even in soil just outside my lab window over here. We found these genomes, but we still don't know what they infect. We think, actually, to kind of that whole full circle we talked about at the beginning, is microbial, we've got bacteria, we've got archaea, but we also have microbial eukaryotes. We think that these viruses are actually infecting microbial eukaryotes. So yeah, we grow E. coli in our lab, we grow Saccharolobus in our lab, and we're now growing some eukaryotic microbes in our lab as well. So we're covering all three domains of life in our little Extreme Virus Lab at Portland State University.
Wow. Yeah, I've really been interested in the micro eukaryotes, for lack of a better word, for quite some time. And this makes me think about the expression microbial dark matter. And a lot of people don't like it, but I've always wondered about what we're missing, because almost certainly we are. How does this connect to the RNA world idea?
Oh, sorry, I totally spaced on that RNA world question.
It's all right.
My bad. RNA world idea is that if you think about, and particularly a lot of this comes from viruses, is how did life arise on Earth and maybe somewhere else? How did life arise? And so one of the things that we now know is that we have RNA and DNA, which can serve as genetic material. Now, one of the really neat things about RNA that happens with DNA as well, but RNA has this fun little extra OH on it, going back to my biochemical roots here. And because it's got this fun little extra OH on it, it has much more chemical activity than DNA does. And so the idea here, and I forget who came up with these hypotheses originally, some of the origins of life people, said that, well, you need to have some kind of genetic material for life, and you also have to have some kind of metabolic material for life, something that can perform chemistry. And so the idea was, well, if we now use RNA can have genetic material, which can then get transferred from generation to generation, but also that RNA can have enzymatic activity for doing metabolism. And one of the things that many origins of life researchers have been looking for is the sort of holy grail of the RNA world is you have a RNA-dependent RNA polymerase, which is made of RNA. And so what that means is you have RNA, which is your genetic material, but then you have an RNA which can copy that genetic material. So basically your Rube Goldberg machine, which keeps copying itself over and over. And so that's one of the big central ideas of the RNA world hypothesis. And a number of people think, including myself, is that potentially some of these viruses, which are still using RNA as the genetic material, like everyone's least favorite virus these days, I think SARS-CoV-2, use RNA as its genetic material. So maybe these are holdovers from some kind of RNA world where RNA was being used as the genetic machinery. Now, we all use DNA, right? So how do we go from an RNA world to a DNA world? Well, maybe something like these Cruy viruses, where RNA and DNA actually got together, could be a way that RNA world has moved to DNA world. This is highly speculative, of course, but really fun to speculate about.
Oh, no. And I've watched, I make a joke of this, and I think you've heard me say this, is that science progresses through a series of steps. And I think this is from Haldane originally, that it's a silly idea and you're foolish for bringing it up. Stage 2 is it might work a little bit, but it's not relevant to the natural world. And stage 3 is I said it was a good idea all along. And then one of my research students years ago, Micheline Wong in California, came up with the fourth one, and I came up with the idea first. and you're laughing because we've all seen it.
Absolutely.
And so this idea that RNA can have catalytic activity, of course, I've watched this process going on and on and on with it. And when you look at the very ribosome itself, I mean, the peptidyl transferase is an RNA, catalytic RNA, a ribozyme carrying out that function. And people only found that out maybe 15 years ago. So I agree with you that looking for the more arcane and understudied RNA viruses may tell us all kinds of things. Absolutely
agreed. And I'm not the only one who thinks that this is a really interesting thing to be looking at. There are, again, one could do a whole podcast on viruses. In fact, there is one this week in virology. But thinking about some of the what they call viroids, or things that are RNA that are actually not coding for any kind of proteins but still can replicate themselves, there's a fascinating group of, we can call them organisms, we could argue whether they're organisms or not, to look at in terms of understanding some of these processes. And one of the things we also has to always bear in mind, we're looking now at the current level, at the current day. What happened 4 billion years ago? That's much, much, much harder to figure out. And something that, not surprisingly, NASA and other people are thinking about if you're going to be looking for life and origins of life on other planets, such as Mars, Europa, etc.
Have you ever read Graham Smith's book, The 7 Clues to the Origins of Life?
I haven't. I should probably try and check it out.
I will get you a copy. Yeah, I will. Exactly so. We've talked for quite a while, and it's been lovely. I like your energy. I like the detail. I like how personable it is. You make everything super accessible, which is my goal. Remember, these podcasts are as much for me as for the listeners, so I've really enjoyed it. But I did want to ask one final question, if I could.
Please.
What was the one event that sold you on being a microbiologist?
So that's a really tough question to answer. But I would say really, really sold me was when I first went with Wolfram Zillig, one of Wose's army, to the first hot spring where actually Saccharolobus sulphotericus was originally isolated. And we were collecting a sample from that spring. And I just thought to myself, wow, I'm working on something which is real, it's in the environment. Up to that point, I'd really been doing a lot of classic molecular biology, bands on gels or turbid kinds of solutions. I didn't have that immediate connection to the real world, as it were. And that's actually one of the reasons that I try and take as many people in our group to Lassen Volcanic National Park to get that same kind of experience. You're working on something which is there, it's real. And I think that for me was a real connection. Yes, I was really excited about microbiology beforehand, but that was the real kicker, if you want to call it that, in terms of getting me just completely 150% in.
Well, Ken, this has been an awesome segment. I use the term awesome perhaps too much, but it certainly qualifies here. I'm glad to have met Stanley. I believe I've met Stanley before, but Stanley now has a wider viewership.
Absolutely.
And I can't wait to find out the other viral structure, which will be really fascinating. If you ever manage to pull that out, it'll be fascinating to see. Well, Ken, thank you. It's been a wonderful, wonderful session, and I want to thank you for your time and interest.
Thank you, Mark.
This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, comments, and suggestions to me at mattersmicrobial@gmail.com. Show notes from today's session with awesome links can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Ken Stedman can be found in the biology department at Portland State University in lovely Portland, Oregon. Many, many thanks to David Renata for the superb editing and Reber Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum today. See you next week on Matters Microbial.